Do you have YouTube on your phone?
On my phone?
I should be live right now. I don't know how.
What's the channel?
I don't remember now. I'm still learning how to do this, so it's supposed to be just, I'm guessing by my name, but I really don't know. I'll figure it out and post it. I do know that it works though.
I don't know how often you guys are on campus or anything, but have you guys ordered from the little robot?
I have not ordered from Little Robots.
They're so cute.
They're precious and I love them.
I know, every single time I see one, I'm like, oh.
My favorite occurrence was encountering them all being like driven back to Avenue C, or not Avenue C, Auburn-Payne, where like there's a little setup for it, you know. I was being driven by there because I was drunk, so I was being sober driven home, right? And there's a whole little fleet of them.
Yeah.
There were like 10 of them, and I'm just like, oh my god, Evan, look! And he was like, I see them. And I was like, look at them.
So like Thursday night, you know, when they first had them, like they had them all lined up out there and they were escorting them inside one by one. I was like, oh.
Yeah.
At first I didn't know what they were. I was like, what is going on here?
It was precious.
Yeah.
And it evidently does not take a lot to make drunk me happy.
Then
I was like, oh my god, they're fucking cute. I mean, I already knew that fact, but like.
like the other day, I like when there's like multiple ones going behavior all at once and they're all in a little line. Like the other day they were going like between the library and the humanities building. It's kind of rough right there. And they were all going, and so like the first one it like slowed down to go over the bump, and then the next one slowed down. And like it was so cute.
Apparently, if you walk around them in circles, it messes up their GPS and they start spinning.
Oh.
They just go. I got more progress done on my essay. It took 2 energy drinks and going over to one of my friends' apartments, but
yeah, at this point, I'm just gonna have to practice. Like I literally, like I just can't.
this is the first time in my life that I've never waited until the last minute to do like a lot of shit.
Mm-hmm.
Like I turned in the take-home exam Thursday, like last Thursday.
Mm-hmm. Yeah, I did mine Friday, which was early for me, for sure.
Greta and Jacob were texting me on Sunday, and Greta was like, I still haven't started this SMH, and I'm like, Greta, babe, come on. Come on, man. And then Jacob sent me a video of him submitting it at midnight.
Oh.
He just sent me a little video on Snap, and it was him clicking his mouse on the submit button and getting like the confetti, you know?
Uh-huh. And that's probably gonna be me with this paper on my own. Like
I worked the day of the paper, so I fucking can't get it off. Yeah, until last minute. Like I get off at 8:30, and there's not enough time to write the paper.
Yeah. Like I told my boss, I was like, I'm probably gonna have to skip out on training. Like I'm probably gonna have paper to do. I mean, I'm gonna try and get like tomorrow. I will try and get as much done as possible. I don't know, for whatever reason, like when I'm trying to read my sources, I'm just like.
Oh shit! I have a quiz tomorrow. I forgot about that. In biochem. That's wonderful. Also, the due date's been extended for the thingy that I wasn't gonna do. I'm still probably not gonna do it, but
Dude, I wish you would extend the due date.
yeah.
Like, it would have been nice if it would have been due, like, right after Thanksgiving. Thanksgiving, I don't work on it. I mean, I know some people don't like doing that because it's supposed to be a break or whatever.
Yeah, I am glad that it's due before Thanksgiving break, but honestly, I wish that was just the whole paper due before Thanksgiving break and not a rough draft. I fucking hate doing rough drafts.
I do too.
This means I'll have less to do like the last week of class, but like whoop-de-doo, I already added 2 presentations onto my plate. So
Yeah.
the only sign-up slot for senior seminar that I could get was the one on the 30th because like there's still several open during finals week, but like I already told my boss I could work all of finals week. So like, and I now have another new presentation during finals week that I have to worry about. So I have to be like, hey Macy, remember how I said I could work all of finals week? I'm gonna need 3 to 5 p.m. on Monday.
Oh, so you aren't coming to the one that's for senior seminar, or not senior seminar, population.
Well, I was gonna try to
Oh,
if I wasn't scheduled for work, but I emailed Dr. Duberell and I was like, hey, I filled out the when to meet with my final schedule availability, but I have no idea what work's gonna schedule me. I already told them they could put me to work all during finals week because A, I need the money, and B, they need people. So, and he was like, okay, thanks, just let me know. I was like, cool.
I don't think it's that big a deal. I mean,
It's, yeah.
as long as a decent amount of people show up, I guess. Which I'm curious, do you think he'll, because like he said that, you know, the audience was supposed to be grading whoever was presenting. Do you think that's going to be like part of our grade? Or
I don't think so. I know part of the peer review is going to be a bit of our grade for our rough draft.
Yeah.
Like people are going to give us a grade on our paper. But I think for the presentation, it's just going to be like, he might take it like a little bit into account, but like if people say we did really shitty, we didn't, because obviously we'd be like, no, this is dumb. But he might use it for like reflection for us on our speaking abilities. I don't know if you were in Dev's freshwater. Okay. Dev had us fill out little morning pages about people's presentations, like things you like, things you didn't like, or name one thing they could improve, one thing they could, or that they did really well on, and then give like a rating out of 10 for these 4 categories.
Well, see, I'd go the opposite way though. I wouldn't like grade somebody really hard. I'd actually probably go.
They were anonymous.
Yeah, well, but I'm just saying, I would probably go. I mean, I don't know, because I know presentations and stuff are hard, and sometimes you don't have bad days.
Maybe I'm just an asshole then. Because there were some presentations that I was like, ooh, these are not well organized. You could have done better.
Well, I mean, if somebody like really doesn't do what they're supposed to do, like and if it's something like they just, you know, put crap together.
I also really hate it when people have like paragraphs on their slides. I blame Dev on that because Dev is like, you need to have no word or like very few words. That makes sense. Like if you have a lot of words on your thing on the thing.
Well, so like we had to do a senior project in my high school, and that's what we had to do. We had to do like, you know, just a few bullet
For my
points
project,
with
if you remember what I
at
did
most
last
3
year,
words.
how I was trying to isolate cobalt.
So,
This year, I'm thinking
like
instead
that you
of
were
that,
just supposed
what
to
if I
do
find
prompts.
something?
Sorry, what?
Yeah, what if I find an organism where that's close enough? What do you want?
Tell them no.
Yeah, that doesn't sound quite right.
But right about what?
Okay, so I'm
juggling both, setting both, trying to answer your question. So, there's that. Let's see. So you need to refresh my memory on essentially what you're trying to do with your project. So it's to essentially get out specific metals.
Yeah, so he said that the problem is iron makes up like, I don't know, 70+% of the metal concentration in there, and it's useless basically because there's no real use for it in terms of like economic benefit, especially in these instances. But if you can take it out, then you have a solution that's left with zinc, copper, cobalt, and a couple other really trace metals and stuff that would actually be economic to remove. And also they're all not like most of them are all toxic to a certain extent anyways, especially if you leave them in the ground, because that's how you deal with all societies, very basically. So
Okay,
then you
well,
have
if
this
you
solution
can
that
essentially provide rationale
Okay.
and
I got you.
so I mean, you really have to be able to defend your ideas and what you're proposing, why it'll work.
That works.
Yeah. So if you do that.
Well, it will be, don't worry.
I know.
I'm gonna be using your favorite kind of micro stream files. Make some lovely pH solution later too. That time.
Missouri weather really always throwing us for loops. Like, I know part of this is definitely climate change, but part of it's just the Midwest being dumb.
Well, it's not necessarily dumb. What it is when you think about
Incredibly
how
variable and how the
jet stream and all migrates up and down.
I know all the ecology behind it and the climate stuff behind it. I still just like to say the Midwest is dumb.
Now, better watch out because he'll say something like, I'm going to move out of the Midwest and never come back. And then voila, you're back in the Midwest.
And then you realize it really doesn't.
Not that I haven't lived that myself,
You
but,
realize it wasn't much better anywhere else.
huh?
You realize it wasn't much better anywhere else.
Oh, yes, it was. Washington State.
That's where I want to go. That's where I plan on going, actually. Like,
It's cold out there, though.
okay.
Sounds fire.
I mean, I feel like in the Midwest we're like,
Right.
Depends on where you live. And
it depends on
actually,
how high you are. in the
Not really.
mountains, And also, like,
well, if you're in the desert part of Washington
Yeah.
State, which actually I might.
the
Would you
proximity. Go ahead. The proximity to the ocean makes the temperature changes more mild. So it has it has a lesser extreme range than say like the Midwest. It gets way colder here and way hotter here than it would in Washington because how close it is to the ocean. At least you're happier there, right?
Yeah.
Oh,
See, that's on the west side of the mountains.
Yep.
Go to the east side of the mountains where you've got desert and it's really amazing just
I
the
love it.
temperature fluctuations.
I really like the West
Especially when
End
I'm sitting in the
Mountains.
middle,
My
down,
cousin
floating
lives out
down the
there.
But
I
at least
miss
you're
him a
not
lot.
so crowded.
river. You think
See,
that things
but
are-
actually.
I grew up in a town with like 300 people, so
and the
honestly,
metal is
I'm ready for a change.
Yeah, that seems about right.
No.
I feel like everyone's like, oh, the weather in Missouri is terrible. It'll never be terrible anywhere else. Well,
It absolutely
it
is.
definitely can be. Yeah.
And at least we're not the Northeast.
Oh, true.
Oh, no.
So I was living in Boston. Boston was wonderful.
It's today the day we draw.
Yes.
Cookies. Oh, she brought us cookies.
No.
That'd be kind of nice.
That would have been cool.
Basically like your.
How do you know if I can even bake cookies?
Nobody said bake. We said brought.
You can buy them in a package, or you can have a pre-made cookie dough that all you have to do is cut it up and throw it into the oven for like 10 minutes.
It's still possible to mess that up. I've seen it happen.
Very excited. Yeah, it takes a lot of ingredients to burn.
Okay, before we get started, graduate students, I need you to come up here and take a little slip of paper. Look at that little slip of paper. Come on, guys.
We all come?
Yes,
I'm gonna
yes,
say,
and also what, see, I learned
keep the bad
So-
fortune.
to make sure and actually mark down
The days.
the days.
I don't want that one.
Okay,
Or that one.
okay.
You get a good one, Brian?
I don't know.
There you go.
Nor do I.
This one.
Oh, number 2. No.
What?
It just has a day on it.
Okay, now, look, hold on to that. Here you go, Jeffrey.
Wednesday, oh. I need to do a lot of work.
Which,
over Thanksgiving,
okay, what
apparently.
date did you get?
The 30th.
30th.
Oh, no.
Okay. Okay, so 30th, first or second?
I finished mine. And I'm going.
I hate you.
Second Wednesday.
Okay, you can keep that.
All right.
Okay, Laura.
Second Monday, December 5th.
December.
Hey, Josh.
Wait, how is this the second Wednesday of November?
No, you're the second on that day.
Oh.
Yes.
How
Josh.
about-
Yes.
You know what this is called?
Monday.
I win.
Okay.
It's called calculation more, clearly.
It's called karma.
Let's see,
This is
Sarah,
paying me back
are
for a
you the
lot of
first
things.
speaker or the second? First, okay. And then, let's see, Brian, were you the first or second?
Second.
Dang.
Okay, Jeffrey, first or second speaker? First. And then Laura, first or second? Second. And then Josh,
It says second.
second, okay. Okay, and then as other students come in, they can draw, or I can randomly draw for them. Ha, ha. So, okay. Oh, speaking of which, Peter, come on up. You need to take and open it up and tell me what it says.
Friday, December 2nd.
December
Second.
2nd, and are you first or second?
First.
First? Okay. Okay. So, yeah, let's make that like that. Wait. I mean, you're over your edge.
Okay.
Okay. So Brian, can you be my timekeeper today? Because Lyndon will be presenting his paper. We're going over the discussion. So, okay. Okay, so, another thing, so it's like I also owe you guys, what we're gonna do about that last exam. So what I'm thinking about doing is providing you with 3 potential assay questions a week before the exam. And for the, it would only be the multiple choice and short answers, and then that, or before the exam, you would select one of the 3 assay questions and send that to me, and I'll set something up on Canvas.
One more
So
time?
we each get
Okay.
to pick the essay question that we
You
do.
each get to pick the assay question out of the 3. That has to be submitted before the exam. And I'll set up a place on Canvas where you can essentially cut and paste your assay question, the question and then your answer. And then you come in and take the multiple choice short answer part.
I like where you're going with this.
That way, you've actually got some time for the assay question. You can get it in order. And then you're also not going to feel incredibly rushed to do the multiple choice and the short answers. You'll have the full 50 minutes, but you don't have to worry about having to quickly write out, an essay that's worth a third of the exam.
Will we have 70% of the exam technically with 50 minutes to do it? Okay,
Correct.
so you're not going to increase the amount of multiple choice and short answer. Okay. So
Yeah,
we'll
see,
be
that
doing.
would be evil.
Okay.
Okay, and that's what other disciplines would, we biologists take pride that we biologists are reasonable.
So we do
Professors.
70% of the exam in class and
In
30%
class,
of it out.
30.
Okay.
Correct. That's it exactly. So, sounds reasonable? Okay.
Josh appreciates it because, his presentation's on the 30th and,
I'm gonna get rolled so bad, holy crap.
you
It's gonna be rough.
But then we're not turning it in.
If you keep this up, Josh.
Hey, it's okay, the rest of the grad students will have a low bar to exceed, so they'll do better. It's okay.
And I appreciate that.
Okay, seriously, the rest of you guys do not listen to him.
You guys will be
Okay,
fine, I'll do
okay.
so bad, it'll make yours look better.
Okay,
right.
any questions, concerns, worries? Okay, barring none, I do want us to get through the biogeochemical cycles, because I can't believe that it's like, Friday. It's the last, actually, no. We are supposed to have 2 other article discussions, and I have been hounding those students to sign up and provide articles, but there'll still be 2 other articles that should be presented. And since the Monday after we come back from Thanksgiving, it's only going to be 1 student presenter. So I'll finish up the Monday after Thanksgiving. But, yeah, so. Okay. So with that, let's talk a bit about lignin. So we had talked about cellulose and hemicellulose, and actually those particular large polymers, organisms have evolved very specific enzymes, cellulases, hemicellulases, to essentially break those compounds up outside of the cell. And if you remember, what's the difference between like an cellulase versus an exocellulase?
the exocellulase is the one that they like, secrete to break it down outside of the cell?
Well, okay, so this is, I don't really mean this to be like a trick question, but it's what it is. So the group that is the enzymes excreted outside, they're all exoenzymes because they're excreted outside of the cell and then they work outside of the cell. But I was trying to be a bit specific and essentially distinguish between the two different cellulases that we discussed last time. The endocellulase, so focused on the structure itself, and exocellulase. You have a clue?
I had something and then your answer to Josh made me think that it's not right. So
Oh, no.
endocellulase is cut inside of the cell, and
Oh,
Yeah.
Okay.
yeah.
Sorry. Okay, so think about, Michael.
Endo cuts like internally the molecule exits between the second and third sugar.
Yes, perfect. Okay, so endocellulase cuts within the cellulose molecule, that large biomolecule. Exocellulose specifically cuts between the second and the third glucose from the end of the cellulose molecule. So, and then with that, when you have those two sugar units, one sugar unit, the cells can then take it up and then it goes into glycolysis. Okay, now lignin is very much different because essentially when we consider the lignin molecule, which is primarily used to provide structure as well as essentially to prevent the biodegradation of plant material. Lignin is made up of these aromatic alcohols. Okay, and so essentially what those aromatic alcohols are is that you essentially have a benzene ring and you have alcohol groups sticking off of it and other moieties. And then the plant produces these monomers, and then it apparently, nobody actually knows for sure, but apparently the plant then randomly produces polymers with these monomeric units. And so what happens is that instead of getting the structure that is very, essentially very standardized, you then get this random just group of these monomeric molecules to form this very complex polymer. And so what happens is, considering evolution and all, it's very hard for organisms to produce or essentially evolve to an enzyme that's going to be specific to being able to degrade this very random polymer. So you had your hand up,
So
Josh.
lignin just a grab bag term for these polymers that are like never the same.
So,
But it's not cellulose or hemicellulose.
right, which tends to be very regular. And so, let's see, so we have the two aromatic. Okay, polypropylene. Yep, so this says there. And so what organisms have to rely on are non-specific enzymes. So, and these non-specific enzymes actually tend to form oxygen radicals. And the oxygen radicals is what breaks up the bonds between the monomers. Yeah, Peter?
Yeah, so I was looking at this heterogeneous polymer. What about,
Uh-huh.
it slows degradation, right? What about the homogeneous?
Okay, so when we're talking about homogeneous molecules, that enables enzymes to evolve over time to be highly specific and thereby are going to have faster rates of reaction. But with these, it'll still happen rapidly, but what you're doing is you're relying on the formation of oxygen radicals. And then when you think about it, with the production of oxygen radicals, that's not going to necessarily be so good for the cell itself. But it is a way that if it has repair mechanisms to be able to get a source of carbon. Okay, so when we do consider, essentially lignin degradation, we need those extracellular enzymes again. And like I said, they're going to be mainly, like they said on the slide before, the peroxidases. They're going to, another common group of enzymes are lactases. And so essentially what they do is they're going to produce oxygen-free radicals. And then once you get these broken down, so like here, this is ferulic acid, you have your aromatic ring, as well as carboxyl groups and different things. And so essentially, these then, once you get down to the monomers, it can then be channeled into central metabolism. So, let's see, possible ways for human materials to be formed. So this is going the opposite way, essentially having complex monomers, other biomolecules from cells, polymerization. I think we talked about this before when we talked about soil formation. Okay, let's turn our attention now to methane and the different organisms that either produce methane or are capable of metabolizing methane. So when we're actually talking about the formation of methane, okay, those organisms, what compound are these organisms utilizing as a terminal electron acceptor with the formation of methane? What's the terminal? Right, carbon dioxide. Okay, so methanogenesis is actually a very specialized group of organisms capable of using carbon dioxide as their terminal electron acceptor with the production of methane as the reduced product. So methanogens. Now, when we have organisms that are capable of utilizing methane, okay, those organisms that can metabolize oxidized methane are methanotrophs. Okay. So methanotrophs tend to be strictly aerobic organisms. And so they're capable of essentially oxidizing the methane and getting out those electrons to be utilized for metabolism. Now, there's a number, and we'll see a chart in just a moment, but there's a number of different C1 compounds out there. Okay, so methanol is an example of a C1 compound, formate. And so organisms that can metabolize C1 compounds besides methane are methylotrophs. So
they're capable of breaking down methanol despite the fact that methanol is toxic to just about everything that's alive.
So they're microorganisms. Microbes are, let's face it.
Microorganisms.
Right. Yep. So they're capable of, well, when you think about what is it, well, for us, the reason why methanol is toxic to us is when if a person ingests methanol, it's going to be your liver with your oxygenase molecules or enzymes. And what happens is the methanol is oxidized to formaldehyde. And that's why that's a problem for humans. But for these microorganisms, um, what leads to toxicity of methanol and ethanol, because this is a big question and actually environmentally microbe-related, when people are attempting to genetically modify organisms to produce more ethanol, either as something to drink or more likely a fuel, what happens is that the ethanol can start dissolving cell membranes. And so it's the toxicity comes in with just the disruption of cell structure. So that's why you can have organisms. Did I answer your question? Why you can have microorganisms?
The 0.1% against any type of kill.
Okay, so, now when we talk about, and again, as was mentioned before, we really do have to be concerned about methane emissions because methane is such a toxic, or not toxic, but powerful greenhouse gas. And so, when we consider the input from things such as cows, ruminants, termites produce quite a bit of methane because of the gut microorganisms. Rice paddy fields, natural wetlands, landfills can contribute to this. The larger landfills are going to have systems to collect methane that's formed. And then other things to think about is abiogenic. And so actually these numbers probably should be updated because as people are finding with satellite data and scanning for methane emissions, actually old areas where they have done drilling before, the drill heads have deteriorated and are capable of releasing great quantities of methane. So, yeah.
I missed the definition of ruminants. What would you define
Oh,
ruminants as?
Okay, so ruminants are those animals that have like extra stomachs.
Like they're on cows and
Yeah.
cows. Okay.
So,
I thought that would have been higher because people complain about them being one of the primary sources.
okay, so what you have to always remember is that, there's data and then there's people's perceptions
Okay, so
of
their
the data.
perception is that it's much higher as opposed to the
Right, because you can see cows in the field, but you don't necessarily see the methane coming up, from these other sources.
I see.
Welcome. Although, natural wetlands, so one of the things that Dr. Westenberg has done in the past is the Voltaire experiment. The Voltaire experiment is when you go to a wetland and essentially you take a big funnel or a garbage bag or something, but what you do is you essentially go to one of these environments, poke around, there's gonna be lots of bubbles that come up, those bubbles are methane and carbon dioxide. You trap that, and you have to do this at night. And then as you're releasing the gas, you light it, and it's actually pretty cool. So, but don't do it. So, okay.
Dr. Moore, I have a question.
Yeah.
A lot of landfills have these things where they have gas in the pipes or the methane, and then oftentimes they end up, like, especially around some of the ones that I live by, they would light them so that they would
There's a
burn
burn
off.
off, right?
So would that help control the methane release then because they're burning it?
Right, because essentially what they're doing in that, and in fact, with some of the, and you notice this actually as you drive through like Illinois where they're pumping crude oil and all too. It's process of flaring. And so again, since methane is a more powerful greenhouse gas than carbon dioxide, it's better to flare it, burn it, convert it to CO2. So that's what they're doing, both with some of those landfills as well as, drilling for crude oil. Okay, so I mentioned the C1 compounds. Okay, so this is just a list of the C1 compounds. And so it's the methylotrophs that are capable of essentially utilizing these compounds as a source of carbon. And for some of them, it's also going to be a source of hydrogen, such as formate. Service is used as a source of electrons because of the hydrogen. So, okay, so let's go ahead and now move on to nitrogen. So let's consider the different reservoirs of where nitrogen is. And actually, the majority of nitrogen is in our atmosphere. Okay, now with the nitrogen in our atmosphere, okay, essentially, I would actually, can argue that it is slightly recycled, but it's just at very, very low concentrations. Okay, and one of the things that we're going to be talking about in just a little bit is nitrogen fixation. So the ability of microorganisms to take dinitrogen gas and essentially form ammonia from it. Let's see. So other reservoirs, again, the Earth's crust. So formation of rock that contains nitrogen. And then when we consider like the rates of processing through these different reservoirs, okay, biota, living organisms are going to be cycling through it quite quickly.
So the three highest concentration things, the atmospheric nitrogen gas, the soluble nitrate salts, and then the Earth's crust are all not actively cycled.
Okay, so like I said, I would argue that the
With the exception of this very slow process of the nitrogen in the atmosphere being processed.
Mhm.
But
Right.
For the most part, they're not actively cycled.
Right. Yep. And so one of the things that this has actually led to, when you think about it, and we'll talk about this a little bit when we talk about nitrogen fixation, but one of the huge innovations when people were, starting farming and everything, and then progressing to your essentially being able to take dinitrogen gas and essentially form usable fertilizer from that. So, but it's also caused a lot of pollution issues and all too. So now we'll go through the different parts of this cycle, but essentially what it really comes down to is we are going to mainly focus on the mineral forms of nitrogen. When we actually talk about the biological organic forms, which is what's, rapidly recycled, essentially it's going to be going from the most oxidized form of nitrogen, okay, which is nitrate, to the most reduced form, ammonia, as well as elemental nitrogen. So when we talk about nitrogen fixation, okay, so again, we're really focused on taking dinitrogen gas and having ammonia form from it. Now, why do you think that this is such a difficult conversion? Okay, so abiotically, to produce not fertilizer, you have to have high temperatures, high pressures to form fertilizer. But these microorganisms are doing it actually, well, probably not in the soil right now, but if it was warmer, there's probably nitrogen fixation going on.
The nitrogen triple bond is one of the strongest bonds.
Yes. Yep. So the reason why this process takes so much energy, and actually it doesn't say it on the slide, but when you are converting one molecule dinitrogen gas to ammonia, the cells have to use 16 ATPs and a bunch of hydrogens, a bunch of electrons. Now, essentially, it's very energy and electron intensive. So, let's see. So, when we talk about the nitrogen that's fixed in the industrial processes, it's a Bosch process. Again, it requires very, very high temperatures and pressures, so it's very energy intensive. And so, with microbial nitrogen fixation, this can actually be done by free living organisms or through symbioses in root nodules. And so, like Dr. Westenberg studies, is he specifically studies rhizobia, okay? And these are going to be the nitrogen fixating organisms associated with legumes. Legumes are typically bean, pea plants that can form nodules. But then you also have free living organisms. So Frankia is an example, and okay. This is just telling you some of the where most of the nitrogen fixation occurs. So essentially, quite a bit of it is going to be due to man-made fertilizer. So there are some representatives of free living nitrogen fixers. So now one of the things I do want to point out, so there are strictly anaerobic, and then there's strictly aerobic organisms as well as facultative. So this process of nitrogen fixation actually has to be done under anaerobic conditions, because the actual enzyme that actually has molybdenum, and you don't need to remember that, but it has a metal cofactor, molybdenum, which is very sensitive to oxygen. And once oxygen is introduced, the enzyme is rendered inactive. Yeah, Josh.
This is where we get heterocysts from, right?
Huh?
This is where heterocysts
Yep.
come into play.
So you're jumping ahead. Yep, but that's fine. Okay, so essentially, even when you do have these strict aerobic organisms, okay, so in what you're talking about with the heterocysts are going to be the cyanobacteria. These organisms form specialized cells with very heavy cell walls that prevents the introduction of oxygen to where those enzymes are. Now, for the organisms that Dr. Westenberg studies, so the aerobic organisms, essentially the reason why the symbiosis occurs between the plants and these bacteria is that those nodules form, and the structure itself helps prevent the intrusion of oxygen, but they also produce this material called leg hemoglobin. And the leg hemoglobin is similar to our hemoglobin, And it specifically binds oxygen and keeps it away from the enzyme to fix nitrogen. So, let's see. And this next one just shows the different rates of fixation. And then what we'll do next time is talk about nitrification, but just to jump ahead a little bit, with nitrification, it's 2 steps, and this is critical for wastewater treatment. So, okay, so that is here. And Evan, it is all yours.
So my name is Evan. I'm a junior here in the biological science department with a pre-med emphasis. So this is environmental microbiology is not my specialty, so have some pity, please. So also forgive me if I talk a little slower or take a lot of breaths. I'm dealing with mental stuff. So. All right, so this article that I picked has a ridiculously long title, and you'd think they could make up something better after all the hours of research they poured into it. But I suppose not. Anyway, so getting right into the abstract here. So this is a relatively recent study. It was just done, I believe, in 2016. And the name, while it might be a little excessive, kind of canvasses the point perfectly. So the whole point of this study is to determine whether or not high nutrient concentration can induce the virulence factor expression and cause higher virulence in an environmentally transmitted pathogen. And so firstly here, the abstract gets into talking about the different environments of one of these environmentally transmitted opportunistic pathogens, and that is inside the host and outside the host. And one of the key differences here is nutrient availability, and this really stark contrast between nutrient levels causes changes in the pathogen's behavior. But something that's not yet understood before the study was the effect of nutrient levels on the virulence of these pathogens. And this article is all about the pathogen Flavobacterium columnar, which is an ecologically important fish pathogen. This bacteria was cultivated. So to do this study, this bacteria was cultivated in cultures with varying nutrient concentrations. Expression of genes that altered virulence was measured with reverse transcription polymerase chain reaction. And then the bacteria are put kind of to the ultimate test in how well they fare against rainbow trout fry, which fry is a term for young fish. I did not know, but so the introduction here has some brief background that's worth diving into. Is that this pathogen is of particular interest. It has caused innumerable amounts of economic damage to the freshwater aquaculture industry. And one of the reasons for this is that it is able to persist for very extended periods of time outside of the host. So it does not need a host to thrive and live on its own. And despite its ecological importance, its mechanisms of virulence are still relatively unknown. And then just one more time right here to reiterate, the whole point of the study is to identify the outside host conditions that have an effect on the virulence of this pathogen. And so, and something that got to write down here that I really thought was worth mentioning was that there are two enzymes that play a key role in the virulence of this pathogen. And the names of the enzymes are chondroitinase and collagenase. And these are tissue degrading enzymes. So the symptoms of this pathogen will oftentimes be seen in skin erosion, lesions on the fish itself. And so getting into the first figure here, we kind of have three different morphotypes or colony types: rhizoid, rough, and then soft. And these three different colony types will exhibit these two enzymes at varying levels. And with this particular graph, this was all done in a liquid culture. And as can be seen in both, even just by a little bit here, chondroitinase and collagenase are both exhibited best on the rhizoid morphotype in a liquid culture. But then it's seen a little differently here. This is on an agar plate culture. You see chondroitinase is still grown best on that rhizoid morphotype, but what's interesting here is that collagenase is best expressed on the rough morphotype. And what's interesting about that is that the rough morphotype of this particular bacterium is not virulent, whereas the rhizoid is. And this table, I mean, it kind of tabulates everything with the graphs shown. So I mean, yeah, then jumping to these figures here, these are this is the really interesting stuff right here. So these are both the rhizoid morphotype of two different strains of the same bacterium. And as you can see here, and well, firstly, these denote the nutrient concentration of the cultures along with the control. The x-axis denoting the time and hours allotted up to 26, and then the survival percentage is